Hi everyone, this is Mary from the European Pain Federation EFIC. Today I'm joined by two representatives from the IMI-PainCare Research Consortium to discuss the BioPain sub-project. So Umbretta and Tony, thank you for giving up time to do this interview this morning. Could you start by introducing yourselves, please? So Umbretta, you can go first.
Thank you for the invitation, and I'm Umbretta Caspani. I work at the University of Heidelberg as a scientist and a scientific project manager, and I am the deputy coordinator of the IMI-PainCare as well as of its project BioPain.
Great.
So, Tony?
Hi, I'm Tony Blockhill. I'm an Eli Lilly funded research fellow at the University of Bristol. And as part of the BioPain project, I co-lead the effort to take the experimental approaches the consortium are using in humans and applying them in a preclinical setting.
Great. So Umbretta, can you briefly tell us about the BioPain project?
Yes. So BioPain stands for biomarker of pain, which are these objective measures of pain. And so when we think about pain, we have two components. One is the individual perception of pain, and one are this activity, this electrical signal that are happening in the nociceptive system. With BioPain, we measure the individual perception of pain using patient-reported outcome measurement. And also, we really apply this technology to measure signals in the different compartments of the nociceptive system. We do that in human as well as in animals. We use techniques like EEG and fMRI and spinal reflexes to target the central nervous system, and we use threshold tracking to do measures in the peripheral nerves. And we collect all these things with the final aims of better understanding how pain works and of improving the pain research and refine these technologies, the application of these technologies in the pain.
Great. And Tony, in your view, how may BioPain help clinicians and patients with pain in the future?
Well, we know that painful health conditions are one of the leading causes of disability across the globe. And one of the factors that contribute to this burden is the fact that current therapeutics are often not fully efficacious in patients, and they're accompanied by a range of negative side effects. So we consequently require new pharmacotherapies to better meet the needs of patients and widen the treatment options available to clinicians. And one focus of this project really is to provide the tools, the biomarkers that Umbretta mentioned, and allow those to be applied to the development of novel analgesics. Historically, there have been many failures in development. Lots of drugs that have appeared promising preclinically have subsequently failed to show efficacy when ultimately tested in humans. And there are multiple reasons why this is the case. But we know through the use of these biomarkers that we're using in this project that we can test to ensure that novel drugs are able to modulate the pain pathway in a meaningful way. And this is really important to ensure initially, before we really get into pain populations themselves, just to check that the drug is modulating the pain system in a way we would predict. And we know from studies that have shown when these types of measures are used in pharmacology studies, that the drug development process has a higher chance of success. So by testing that the drug is doing what we expect in patients to the nervous system, it's more likely to be efficacious in pain and relieve the pain in patients. And another use of these pain biomarkers is in potentially providing a more personalized medicine to specific pain patients. So even within a given disease state, the biological mechanisms that are causing pain in a patient may be different from another patient who's been diagnosed with the same disease. Potentially through the use of biomarkers, we can really pin down a specific patient's biological mechanisms that are causing the pain, and then clinicians will be able to use the most appropriate pain treatment that targets that specific mechanism.
Great, that sounds excellent. And Umbretta, to get to Tony's next step, what are the next steps in the BioPain project to get it to the next level?
Yeah, actually, we are in the last step of collecting data. And so soon we will start the second part, the very exciting and very demanding part of analyzing and making data and the modeling and looking at the result and starting the interpretations. So at the University of Heidelberg, where we host all the data, we are getting ready for this part of the project. And we are really intensely working with all different teams inside in the BioPain, and to prepare the complex database, and also preparing with the data programming plan and all the other things that we'll have also with the modeling and the analysis.
So finally, the last question, this is for both of you to answer. So in one sentence, what would you like future researchers to say about the impact of your work? So just one sentence.
I think it would be great if we were able to demonstrate the benefits of utilising objective biomarkers of nociceptive system function in studies of novel analgesics to demonstrate that these drugs are having a relevant biological effect, and ultimately this will assist in the development of novel analgesics and increase the options available to patients.
Super. And Umbretta?
Yeah, so actually I would really like that they see our work as a pioneering and groundbreaking study that really kick off the use of this biomarker in the pain research, but more important in the pain medicine, and going to the direction of the personalized medicine. And I mean, there is the need, we have the technologies, and we should use them.
Great, so brilliant. That was a great overview of the project. So thanks again for joining the interview and for telling us about BioPain, and best of luck with the next steps in the analysis. So it's all very exciting. So thanks again.
Thank you very much.
Thank you.